Hello everyone. Today we're going to discuss gel electrophoresis. It's an extension of last week's lab, which was an extension of the week before that. So in the first week, we did primer analysis, where we actually identified two primers that we're going to use for polymerase chain reaction, which was used to amplify 4 samples. The 4 samples that we amplified was the wild type, WACA55, WACA22, and WACA8. Now these 3 WACA genes is going to be compared to the wild type, so we could know which one is a bigger deletion, a smaller deletion, and an insertion. So how we would do that is because we actually know the size of our wee gene, the amount of nucleotides in our wee gene. So by reasoning, if it's smaller, or if the fragment or nucleotide amount, the amount of nucleotides is smaller, that would indicate there's a deletion. If the amount of nucleotides in our sample is bigger, that would indicate an insertion when compared to the wild type. And gel electrophoresis this week will help us do just that. It will help us to identify our 4 samples. So it's going to be used to detect the presence of DNA. It's going to be used to give an estimation of the actual number of nucleotides in our DNA fragment. And the way it does that is because we actually know the number of nucleotides in each gene. It's going to help us identify the genes in our sample, or the amount of nucleotides in our sample. Before we actually even start our lab, we're going to need specific reagents. And we're going to need, of course, the 4 samples that we amplified last week, wild type, and 3 other WACA genes, which is basically translated into the wee genes, but we don't know which of these wee genes are a deletion or which of them are an insertion. So that is what we're going to identify. We're going to be using our samples and comparing it to a standard ladder, which is going to be a 1 kilobase pair ladder. And that 1 kilobase pair ladder actually has measurements or calibrations where we can compare or see how, or the size of our gene, or the amount of nucleotide, the amount of nucleotides we have in our sample. So in addition to the ladder, we're also going to need a gel, which is an agarose gel, which was already created before the class started. That agarose gel, we're going to insert the samples in specific wells, which we'll talk about a little bit later. And the agarose gel contains, it's like a sieve. So it basically, I look at it, think of hurdles when a track runner is running hurdles. So the hurdles are basically like the sieve, and the bigger the components, or the bigger the sample, the slower it would run through the gel, because the hurdles would keep it back. But the more agile, the smaller the sample, the smaller the hurdler, the faster it would run through the gel, right? Because it will be able to get through or get over the hurdles faster. So we'll also need a dye. The dye will be used for our samples. We will dilute our samples to 1x. We'll get a 5x dye, and that will be used to dilute our samples. So we'll be using 1 microliter of our sample to 5 microliters of our actual dye. So that dye will help us visualize the movement of our samples as it travels through the gel. So it gives it color, basically, as it weighs down the DNA. But our ladder does not need a dye, a loading dye, because it already has a tracking dye inside of it, right? But our samples will need that loading dye. So what actually helps us see the DNA with that dye in the gel is ethidium bromide. Ethidium bromide basically intercalates between the DNA, the major groove in the DNA. So actually it intercalates, that major groove is where the hydrophobic part of the DNA is. So the ethidium bromide will actually get within that major groove. And because of its interaction in that major groove, it can actually cause problems with replication and transcription, and that is actually what would cause frameshift mutation. So when using ethidium bromide, or actually this entire lab, it's very important to use gloves, because ethidium bromide could be could be detrimental. So what actually, when we make our gel and we have all our samples ready, before we actually insert our sample inside of our gel, we will need a buffer. So that buffer is basically what deprotonates the DNA and helps that DNA dissolve in solution so it will be able to move through that gel. So we'll be using TBE, which is tris, boric acid, and EDTA. That is already also made before the class. So how does DNA move through the gel? Well, DNA is negatively charged, and the reason why it's negatively charged is because of that phosphate group, which we could see it's circled in red. Now that phosphate group here, actually the oxygen around the phosphate would give that DNA molecule a negative charge. So as it's negatively charged in B in our diagram here, DNA would move from a more negative to a more positive side based on the electrodes that are connected as we create voltage or electricity, an electric current through our gel. So DNA would move from a more negative to a more positive side. So the more negative side is where we would load our samples. We'd load it inside of specific wells created by a comb that is inserted inside of the gel from the beginning of the experiment. That comb will be removed, and our diluted samples, our samples would be put inside of our dye, of course, and we would insert it inside specific wells or respective wells. On the extreme right, we could see here is a ladder. That is a 10 kilo base pair ladder, and that will also be used for one of the wells because that's what we're going to compare our wild type and of course the rest of our samples to in order for us to realize which sample is actually, or which Waka sample is actually which we deletion or we insertion. So on our extreme left, which we see in diagram A here, we see 1x TAE. That is just another buffer. It's just like TBE. We see a voltage meter, which we're going to actually use to connect our electrodes, and we're going to turn up the electricity to allow an electric current to flow through the gel. The black part would be the more negative part, which is where the DNA would be inserted, and the red part, which is the more positive part, is where the DNA would run to. Right. So as we said before, the bigger fragment, the insertions, would be higher up in the gel, and the smaller fragments, which are the deletions, would be lower down in the gel. After the gel has run for about 35 to 45 minutes, where we see the fastest running or the brightest part of the gel, which is where basically the dye is, and we're satisfied with how far it runs, we will then extract our gel again using gloves, and we will image it. We're going to use the Lycor imaging system upstairs. Now, in your protocol, I know it says that we would use in the protocol it says that we would use UV ultraviolet light, and we'd have to be very careful as to not look into that ultraviolet light. That is correct. But in the Lycor system, that ultraviolet light is encased within side of a machine, so we will not have to even interfere with the ultraviolet rays, right? So that will be taken care of. And then we image our actual gel, and it will look a little something like this. Now, all these bands, this is a mock gel, so all these bands, these are not bands that we will actually see, but it's a mock gel and shows our ladder, which is in the middle. The cartoon of a ladder is actually on the extreme left, which we see 1 kilo base pair plus DNA ladder. So this is more than 1 kilo base pair. So this is 12,000 bases or 12 kilo base pairs. And then we see 5,000 bases or 5 base pairs, etc. So we're going to use that cartoon or that image of the standard ladder to compare our ladder that we actually run through the gel. And when we compare our ladder, we could actually compare our indentations or our measurements to the actual standard ladder's measurements, and the actual samples that we see running in the middle here will be compared to that ladder, so we could tell the actual size of our nucleotide sequence or the actual size of our gene. So after we do our estimations of the actual size of a gene, it's possible to write our lab report. So the lab report will include what we've covered within the past 3 weeks. So from the primer design worksheet, next to the actual PCR or polymerase chain reaction to gel electrophoresis. So you have to write class has to write a thorough lab report that covers the past 3 weeks labs, of course. And I would like you to follow the rubric as is described in the syllabus. And here's a little hint. The primers have to be as part of your results. Just the 2 primers that you selected, right, will be part of the results. And for that you selected for PCR amplification, of course. And also part of the results, we need the cartoon ladder that we see on the extreme left here, and also our actual image of our gel, right? And in your discussion, you should explain these results and the significance of these results. So we have 1 week to actually write this lab report. Is due on the week of the 27th. So that Tuesday night in February at 11:59 is when our lab is due. Okay, I am going to actually run the gel and post these images as early as tomorrow, Thursday, so we will have it available to everybody so you would be able to write your thorough lab report. Thank you.